So Peter's here today from Casebia, is that right how to say
Casebia,
it? Yeah,
yeah,
Casebia Therapeutics, which was set up in 2016 as a 50-50 split between Bayer and CRISPR Therapeutics, so has a European origin. It's now, you can see where they have offices all around the world, but a couple in the US, but you're based in San Francisco, I believe.
Right,
So Peter has a long history in pharma. He worked for Bayer for many years before deciding to move more into the biotech space. And so welcome. So the company has been set up to draw on the CRISPR-Cas technology and to look into how this technology can be used to help with human health. So you can just see a little bit of their pipeline here, but I'm sure Peter will tell us a little bit more about that in a bit. So the company's setup was quite unusual. I wonder if you can tell us a bit more about that.
Yeah, I can do so, definitely. So thank you, Helen, first of all, to being here. I know actually Philip since 2014 when Labiotech was started. We met at the Bio in San Diego. Remember that, Philip?
Yeah, I did.
After party. So very interesting. And CRISPR, I mean, I came into contact with CRISPR in 2013 the first time. That's actually when Versant Ventures, who's one of the investors in CRISPR, was pitching to large pharma companies getting interested in CRISPR. And guess what? None of the big pharma companies was interested. Not the people itself, but management, I should say. So then in 2015, Bayer founded the so-called Bayer Life Science Center, which is a dedicated function with a lot of funding that can be used for these venture investments. New technologies, breakthrough technologies that have a use for all areas of Bayer. And CRISPR is one of the best examples for this. So you could use CRISPR for therapeutics, so that's the part that we are dealing with. But you could also use it for crop, seeds, and so on. So Bayer has a large crop science unit. So all of that, animal health, assay development for drug discovery, and so on. And so that's why Bayer decided this is the first technology we will invest in. And we were looking, I was part of that team then, looking for a potential partner. So we looked at all the CRISPR companies, CRISPR technology companies, I should say, because the name CRISPR. And then various reasons, I mean, both partners for sure have to agree, and then portfolio, what is available, and so on. We partnered with CRISPR Therapeutics, and it was set up as a joint venture. And Bayer invested 300 million into the joint venture. They also invested into CRISPR Therapeutics. So this gave us a head start with money so that we don't have to think about financing the next round all the time, but we can form an independent company which should, based on the size, create 3 to 5 INDs in a time frame of about 5 years. That was the kind of concept, really with a goal to deliver INDs. There was not a named money return goal. It was really the 3 to 5 INDs. That's our goal to create. And it's a carve out of indications. You saw that earlier, some of the indications. So there's exclusivity for these indications that we work in, and then there's others that CRISPR Therapeutics works in. There's kind of like no overlap. There's one exception, which is in hematology, because they have this Vertex collaboration that you might have heard of, where they are filing, have filed the IND, and CTA. And that's in sickle cell disease, beta thalassemia, that is with Vertex, and CRISPR is not with Bayer, or with Casebia in this case. But other indications are. And to the locations maybe, so the reason for San Francisco and Cambridge is in Cambridge, Casebia is co-located with CRISPR Therapeutics in the same building. And in San Francisco, it's co-located with Bayer. They have a small research site there. So we're in the same building as Bayer there. Seattle Children's Research Institute, that's a collaboration on engineered Tregs, regulatory T cells. Yeah. And the Cologne is actually, that's the Bayer site in Cologne, where we have a protein engineering group working for us. And we will come back to that, I guess, what is the future of CRISPR actually. They are working on optimizing the CRISPR-Cas9, the tools that you use for really doing the cutting and everything. So that's done in Cologne.
So, I mean, the setup, I mean, there are famously a number of CRISPR companies out there, including Editas and Intelligent Therapeutics. I just wonder, can you shed any light on why you chose to partner with CRISPR Therapeutics?
So I think part of that was, I mean, I can tell some, but it was a full matrix. I mean, it was
I'm
like
sure.
the nicest picture of Harvey balls that you can think of. All the companies and all the, criteria from social, everything. Yeah, we put in like what collaborations they have, what rights could we get out of that. Part of that was in the indication spaces as well. Where would these companies be willing to give kind of outside, isn't it? They still own 50%. CRISPR still owns 50%, and they have option rights to the product. But still, they gave it away, isn't it? And if you think about the front-runner projects of some of the companies, they were just maybe not willing to, or it would have been more difficult. And then management would have to agree, the investors would have to agree as well. And then on our side, it was various criteria as well. I mean, whom do we want to work with, and what was the best setup in our eyes?
It seems like looking at the share price, that was probably a good decision, but let's hope. Yeah. But so you mentioned the trial that was the recent IND that has been in the news because of the FDA putting a stop on the initial process. Could you talk a bit more about that and sort of what impact that might have and what the reasoning was?
Right. So good. So I can talk about this. I can explain it to you maybe a little bit, but I don't know the reasons for sure because they are not published. So the FDA will pose questions, but they will not be published initially. So the process is in USA for the IND, you apply for the IND, you submit all the documents, and then the FDA has 30 days to ask questions and kind of like put it on hold, if you will, if they have too many questions. Otherwise, it just continues. So while you submit, you're already in the clinic, kind of, verbally spoken. And that's why it's called on hold, but no patient had been treated also, So it's not like some clinical holds that you hear of where a patient dies or whatever. They just didn't even get there yet. And we don't know, is it a lot of questions? What questions? We don't know. So now the FDA has 30 days time to pose the questions to CRISPR Therapeutics. Then they can answer, and then with the answer submitted, again, 30 days, the FDA can decide. And that's what we have to wait for. Usually at that point, in the acceptance or in the approval, if you will, document, they will tell a few things about what the questions were, but it would be pure speculation, and I don't know.
Yeah, fair enough. I mean, because it's sort of such an early stage thing, I guess you wonder if it's perhaps a bit of caution on their part, but so obviously in addition to that, as was also in the news this week, CRISPR's been a very popular topic. I'm sure many of you will have read about the papers that were published in Nature Medicine suggesting that CRISPR might, if you use it in humans, might increase the risk for cancer. I thought, give you the opportunity to talk about that and say what you think about the papers.
It is true. I mean, CRISPR is very popular. So on one hand, I always say it. So it's the first time you can talk to anybody and they know about it, isn't it? Even parents, whatever, you just talk to them. Yeah, I heard about it. I read it in whatever newspaper or magazine, So it's very popular, but at the same time, a little, 1-2 papers in this case, and suddenly Nature works like they collect the papers, although one was already 1 year old, isn't it? But then they publish one day. So, And so these papers came out about the connection between P53, the gene, and cancer and CRISPR overall. So what they did is in a very specific cell type, and that's very important actually. I mean, there was a very specific cell type that they used, and they saw that when you do the double-strand break, so the cutting, which is shown here, where you cut, and then this P53 gene is responsible for creating this repair or initiating this repair mechanism. At the same time, there's other papers that link P53 activation to cancer. So in this study, they never showed that the CRISPR cut or whatever created cancer. This was not shown. It's just a hypothesis that it could be in principle. And then this very specific cell type. And it has been shown previously, so it wasn't really new. So suppression of P53 has been shown to improve the activity, so because then the repair mechanism cannot start, and so you get more of the desired process. So it was, my personal opinion, overblown, but we will for sure, nobody neglects it. We always watch for these potential risks. You know? I mean, that's for sure taken. And if we work in specific cell types, we do all this work.
Yeah. I mean, talking on a broader sense, I mean, it is a very early stage technology. I mean, it's very unusual to have so much investment in an area where it's, you know, they haven't really got any, other than I believe in China, there aren't really much any in human trials yet.
That is true. Yeah.
I mean, what are you doing as a company to try and make sure that the patients are safe and that it's, you know, the techniques are good?
I think one part is working with the FDA, and I guess the clinical hold is one example where CRISPR for sure will work very closely with the FDA, solving all the concerns that there might be, which we don't know. But also, for example, the other paper that last year came out about the off-target effects you might have seen, so where the CRISPR system might potentially cut at sites where you don't want this. Yeah? Going back to the comparison to the word editor, you're changing the wrong word somewhere else in the text, which makes suddenly bad meaning or whatever, isn't it? So that's potential. But that's addressed, and that paper was retracted actually, so there were several flaws in that paper. Again, stock market went down. I mean, you wake up in San Francisco, stock is already down, you know what's going on. And it's always interesting days because everybody you talk on the phone knows about it already.
Yeah.
Whoever you talk in the world, everybody knows it. It's very fascinating how many people read these news.
Yeah.
Maybe due to
Yeah. Yeah, well, of course, that's what we'd like to think.
But so there's in silico tests and there's other tests in vitro and so on where we check the off-target effects and so on. And then it's all about the specificity as well. That's the Cologne group, as I mentioned earlier, the protein engineering that is improving the cutting machinery, if you will, that it is more selective, specific to the site that you want. And what comes in is the organ where you cut. So you deliver, if you do it in vivo, so there's ex vivo and in vivo possibilities. If you do it in vivo, then you need to deliver the whole system somehow. And that's what we're trying for sure to make very selectively to the organ, maybe even the cells. If you think about the liver, hemophilia, there's specific cells that we want to address where we want to do the cutting. And that's helping as well for preventing off-target effects. So there's a lot of things done, I guess. And then it will be, as I said, working with the FDA, with the EMA, to decide which animal models will be needed to show this. It's very difficult because usually the CRISPR system is animal or species specific. Yeah? So it's not the final product that we would test. That's why they sometimes say you might be able to go ahead without. And I think it will depend on the diseases as well. Yeah? I mean, we have IPEX is not on here. We have published as well that we work on IPEX. That's immune deficiency that we work with Seattle Children's Research Institute. And if those children don't get a bone marrow transplant, they die way early, year 5 or so. SCID, severe combined immune deficiency, they die at age 2 if they don't get it. So there you would, you don't want the risk, but you would go maybe higher risk, potentially, isn't it? At least to save them, and then continuously work to improve if you can. But I mean, if we would know something would create cancer, we would not follow it. Yeah? We would not bring it to the market, I mean, for sure. So.
I mean, I guess in some ways, the fact that CRISPR Therapeutics is going through the trial process at the moment must be very helpful for Casebia, because you're kind of hoping to
we will definitely learn, and I mean, editors also had a delay at least. I mean, it was not a hold or whatever, but they had a delay in their first program. So everybody's learning, and that's good for us as well, because we're still a little bit more away from our first IND or CTA.
So, I mean, I know you can sort of fairly limited to what you can talk about. I mean, what do you think your first product in the clinic will be, if you were looking into the future?
No, so the ones that we have published are our front-runner projects, as you will. So IPEX, I mentioned that already. Hemophilia is one which we are really focusing on, because that's where Bayer as an investor had an interest, because they currently are one of the leaders in hemophilia, and they want to be part of the next generation of a real cure for hemophilia. Currently, these patients treat with recombinant factor VIII up to daily injections, IV injections, not SC or so. And there's more products coming to the market right now that make this easier, but still there will be frequent dosing. And then you have always, as you know, when you dose somebody, you have these trough levels going up and down, isn't it? I mean, the patients maybe forget one time, they're on vacation, there's kids, they don't want, and so on. So with a cure, with CRISPR or gene therapy as well, you would hopefully get a constant level and always be protected during your lifetime, potentially with CRISPR.
That's fantastic.
So, yeah.
I think now's a good opportunity.
Ophthalmology, we're working in ophthalmology as well. We haven't disclosed the specific indications, but those are blindness indications that we're working as well.
Okay, that's really fascinating. Thank you. So I think now's a good time to open it up to the audience. I'm sure everyone's got lots of questions. So who'd like to go first?
So if it's in this area, so what we really learned, I think, what is very important is thinking about the manufacturing already. Yeah? So our key areas are delivery that we're addressing, and very often you go with partnerships as well. And all these companies now, they see dollar signs when they hear CRISPR, isn't it? So it's very difficult for me in business development, but as well, the manufacturing, that you're prepared, because I mean we heard there were some papers about this AV kind of crisis, isn't it? For gene therapy you need the vectors to deliver, and they are difficult to make. You need a lot if you go systemic, and all the manufacturers right now, they are booked out for 1 year, 1 year and a half. So now you have to plan, isn't it? I mean, and then you have to get the materials and so on. I think that's a very important part, that you're not surprised, which is a nice part for us because we're already working with Bayer and so on, so that in case they would opt in at some point, they are prepared, isn't it? And we don't have a break in between, because now suddenly we need to get the material on a larger scale, GMP and so on, isn't it? Certified and so on.
It's a lovely, luxurious position to be able to plan for positive rather than...
That was a great concept of Bayer, I have to say. I mean, very much courage they took for this. Yeah. I mean, it's a lot of money. They had to also, I mean, Gregor is actually a former colleague of me at Bayer. I mean, there's a lot of discussions internally as well, isn't it, about the spending. I mean, why do we give it there, and now we have to cut here and there potentially, isn't it? And so that's happening, yeah, but very good initiative. Yeah. So it's actually, it's in our company presentation. So we work with both AAV vectors, viral vectors, inactivated viral vectors, I should say, and also with lipid nanoparticles or non-viral vectors. Huh? So that's what we're exploring both, and yeah. So it kind of uses, if you will, the guide RNA here as well, but then attached to that is an enzyme that just changes one amino acid. So you're using kind of like the reader, isn't it, when you do the word editing to get there, and then attached to that is this little enzyme which just changes one amino acid to the other. And so if you can do that selectively, I mean, that's just an addition, isn't it, for those where you would need a single correction. So sickle cell disease is actually one of these indications where you have only one letter wrong in your gene, and unfortunately the effect is dramatic. Yeah. So that's potentially, but there's multiple versions that you would need to have all the versatility, so they're not there yet. And you also need to deliver, you need to do the same things, yeah, manufacture and so on. But it's not creating this double strand break in the end. Yeah, so I mean, it's a very good question. I'm not with Bayer anymore, so no. But I was in licensing. I actually worked on gene therapy for hemophilia for Bayer. I licensed that, and we had the same questions for sure. So the market potential is much lower, yes, and that's why, I mean, when you read analysts, for example, for the hemophilia market, they predict 2029 or whatever, 8 billion. That's not true, isn't it? If the gene therapies are coming, that's not true. Yeah? They don't understand this, actually. They think it's 8 billion. And so that is something, but then you have to add more, isn't it? So that's why Bayer said, really, we give you this time, the money to develop the platform as well, isn't it? And with that, we will address more indications. So you could think about other indications. If we solve, that we're working on, delivery to the liver for hemophilia A and all the surroundings, the manufacturing, everything, then we can go to other diseases that originate in the liver. So you add by that. You will have smaller indications, but more potentially. Yeah? And the marketing is completely different, isn't it? So there will be no sales reps running around like Xarelto, the factor Xa inhibitor. It will be completely different. There will be treatment centers potentially. SCID, there's 160 patients globally right now. So, you know, that's completely different model. Yeah? And yeah, same in ophthalmology. These are very scattered indications with small patient populations, because every patient group has a different correction or needs a different correction sometimes. Yeah? So we will enable the platform, hopefully, and then add more indications much more easily. Yeah? It will also involve regulatory potentially, and that they say, no, you don't have to go all the way again, because clinical development is very expensive, isn't it? So maybe there's shortcuts. If you show it's very similar, it's just a different letter that you're changing or whatever, isn't it? I mean, then everything else is the same. Hopefully that will be a faster approval then, yeah.
So how do you think you will address the pricing issue for the sort of hospitals and the providers?
Yeah, so pricing, I think, there are some gene therapies now on the market that have, and also the CAR T treatments that have shown this kind of like high price, if you will, and there's debate, is it justified or not? The Luxturna, which is an ophthalmology product from Spark Therapeutics, where treatment of both eyes costs $850,000. So if you calculate that, actually, it's not expensive, because somebody blind at age 10 or even before, they treat kids at age 1 with this now, because the FDA asked them to. They didn't want to initially, but they were asked to. If you can't learn and so on, I mean, that value is way more than $850,000 to a life, isn't it? So you need some help, a dog maybe or whatever, a guiding dog. But it's a lot of money, and I think with scale, we will also see hopefully some pricing changes then, if there's more indications.
Do you think it might be a bit like with the sequencing revolution, that you initially, genome sequencing was very expensive, and now it's
not that
much
extreme
cheaper.
maybe, but yes. I mean, in general, I think that's expected, that there will be scale effects, definitely. So yes, so there is the Alliance for Regenerative Medicine arm, and we're working with them. We're a member of them as well, and so they're currently working, for example, on creating a kind of lexicon for gene editing, gene therapy, so that the public will understand more or better what this all means. And specifically, if you're a patient as well, I mean, there will be risks with every treatment, isn't it? That's what we know, and you need to understand those risks before saying, yes, I want to do this, or want my kid or to have this done too. So this education is done, but then there's patient groups as well. There's hemophilia. It's a very strong patient population, actually. Sickle cell is a very organized patient population as well.
Yes.
But it's a good point. You have to work early with them, because, I mean, convincing them some of these is not easy. But there's a lot of hesitance in some patient populations because of things that happened in the past, maybe, and then, I mean, they read this as well, isn't it? Like cancer risk, and now you have to argue with them so that they can understand, and that this might not be a risk in this specific treatment that they would get, isn't it? And so,
do you think a way around that is actually working with the patient groups and trying to, inform
so,
them whilst it's being developed, perhaps?
Directly?
Yeah, directly. I mean, yes, definitely.
Without CROs or without working with medical doctors being from hospitals?
No, so you have KOLs, key opinion leaders involved as well. Yes, but I mean, when I was at Bayer, we worked with these directly as well. I went to the conferences. I've seen the patients, and they come to these conferences as well. It's a very sad sometimes, but also very, I mean, gives you a lot of learning. I mean, how these patients, their life and what they challenge. I mean, with the sickle cell, I mean, about the opioid crisis in US, and they don't get the drugs to release the pain, because the doctors say they just want to get the opioid and then they sell it or whatever, and it's not true, and it's very difficult. And you learn all of these only if you interact. Yes, I think, yes, it will be successful. And I want to say, for the ex vivo, there's also a disadvantage potentially. You should compare it to a bone marrow transplant, isn't it? So the patient, you have to condition the patients with drugs so that the immune system is not challenging the new treatment, and also to make space for the new cells, isn't it? And those are pretty strong sometimes. So people are working on improving that, but it's not there yet. So it's a strong treatment, actually, still ex vivo, some of them. Ophthalmology, for example, that will be in vivo, intravitreal injection. So I think there will be some drugs, and Editas is working on that as well. We are working on that. I think there will be something. So that is in vivo. It's not systemic, but localized to the eye, but it's still in vivo. And I think hemophilia, I mean, I really hope that we succeed as well. I mean, otherwise we wouldn't invest the money if we wouldn't believe in it. It's also people working, isn't it? And you don't want to frustrate people, and the patients are waiting. You create expectations. That's actually sometimes what creates the urgency for us to move ahead, isn't it? I mean, people always say, oh yeah, the pharma companies, as fast as possible, they just put it into the market. And no, you get, I mean, people like CEO and so on, they get emails every day, tons of them, from patients asking, when is this ready? When can we have this? I've seen people at conferences, they say, so when can we do this? When is gene therapy available for hemophilia? When can my grandson has hemophilia? When can I, So that's creating the urgency as well. Yeah.
How far are you? Phase 1?
Oh,
Preclinical?
no, we are preclinical, yeah, but yeah.
Well, thank you ever so much. I think if anyone has any further questions, I think you're staying around for some
Yes,
drinks,
yeah,
so
yeah.
please don't hesitate to come and ask. But yeah, thank you again.